Good morning, all. Okay, let us begin. Voice is coming. Let us begin another lesson, that is Sell the Unit of Life. Good morning, all. Am I audible to you? Is everything clean? Hello? Okay, fine. Done. If everything is good and clean, then we will begin in another lesson that is known as Sell the Unit of Life. Unit of Life. Sell unit of life. This is an important one. I will tell you some things about it. Okay. In the past 1 year, from this lesson, out of 90 questions that should be asked in biology part, 14 questions only from this lesson and the associated parts, which means bacteria. Bacteria structure we will study in 2 lessons. One is cell the unit of life. Another one is biological classification. Then altogether from this lesson, it has been noticed there are around 14 questions were there. So with that, what I would like to say, don't leave this part. This one we are studying from 7th class onwards, but this is the time we need to improve our understanding and finally make it the questions that they can ask in NEET exam perfectly answered. Okay, then some parts I will explain and some parts I will ask you to do. So altogether we will make it perfect about the cell, the unit of life. Then come back. First one, cell. What is a cell? Okay, you know what is cell. Tell me what is cell. Generally we believe cell is the basic, structural and fundamental unit of life. That's okay. But in literally, what is a cell? Cell means compartment. Compartment. A compartment or a chamber. That is the literal meaning of a cell. That means like this. You have some body that you are calling it as chamber, or you can call it as compartment, etc., etc. Okay. If you want to make something called chamber, there should be a presence of outer boundary. Outer boundary is what? The inner side, what are the components should be present. All those things gradually we will learn. But keep it in mind, the word cell indicate compartment. Who gave the word cell? Cell term was given by the term cell given by, there was a scientist called Robert Hooke. Robert Hooke. What was the story behind it? One day, he took a cork bend, kept under the microscope, and he observed there are some chambers present inside the cork bend. Cork is nothing but, in the previous class, I think you know, okay, in the anatomy of flowering plants, cork is nothing but the outermost covering that is formed by cork cambium, isn't it? Now altogether, he kept some cork bend inside the microscope that it is developed a kind of chamberized compartments inside. Those compartments he named them as cell. That is the first time cell word has entered in the field of biology. Next one, followed by, there was a scientist called Antony van Leeuwenhoek. This one has improved some studies. He took some drainage water kept under the microscope on a slide. He noticed some bodies are moving in the drainage water, and he called them as animalcules. And first living cell, the first living cell was identified by Antony van Leeuwenhoek. The first living cell was identified by Antony van Leeuwenhoek. Okay. After that, there was another scientist came, Robert Brown. Whether he was really brown or red, we don't know, but his name is Robert Brown. He identified nucleus. Okay. This is the history of cell. How did the cell has come to the field, and etc things, that is history. But before that, one more thing you have to remember. There were some scientists called Hans and Jacarius Johnson, father and son. They were identified simple microscope. Later, this simple microscope developed by Antony van Leeuwenhoek, and recently, right now, we are using something that is called as, okay, electron microscope. Electron microscope. This electron microscope was developed by two scientists called Knoll and Ruska. Knoll and Ruska. These two people developed electron microscope. As the advent of electron microscope, okay, there is an improvement in internal study of cell. If you want to see what is there inside the cell, and the further associated developments developed after the advent, which means when the electron microscope improved, from that moment onwards, the associated developments related to cell developed. Then right now we have something is called cytology. Cytology. What is cytology? Study of cell. Study of cell. One more is there, cell biology. In cell biology, what do we study? Cell and their interactions. How one cell can able to interact with another cell. That we will study in the name of cell biology. Altogether, right now we will discuss about, okay, remember, right now we will discuss about some fundamental overview of a cell. Before that, let us see something that is called as, there was a person called G.N. Ramachandran. Some information given in our NCERT book in the name of introduction of a scientist. That scientist G.N. Ramachandran. G.N. Ramachandran. Gopalaswamy, Narayanaswamy, Ramachandran belongs to Cochin and Kerala right now. This person developed in our Madras IIT one branch that is called Madras School of Conformational Biopolymers. Remember, in the living system, there are many polymeric components are present. Let us take polysaccharides, proteins, nucleic acids. These are all are polymers. These biopolymers will have different variety of conformations that we will study in stereochemistry. These conformations, they are very interesting subject. Over there, if you closely observe the data given in our NCERT book, okay, that data says like this. DNA will have right-handed helices. Proteins present in the living system will also having right-handed helices. That we have studied in our NCERT book. Now you should get the doubt, why only right-handed helices? Because biopolymers are having certain type of appropriate conformations. Those appropriate conformations we can understand by using stereological changes. There was one scientist called Linus Pauling. He explained all secondary structure conformations, but one secondary structure conformation was troubling, that was collagen structure. Collagen structure is very interesting. Collagen is the most abundant biomolecule inside of our animal body. So collagen structurally very difficult to understand. That collagen structure was explained by our Ramachandran by introducing one conformational structure that's called Ramachandran plot. Ramachandran plot. This is a mode of understanding of conformational biological things, a Ramachandran plot. First time in the conformational analysis, some kind of concept was introduced by a scientist, that is our Ramachandran plot. By using the Ramachandran plot, he had discovered triple helical structure of collagen. How does collagen structure develop? Triple helical structure of collagen. The collagen structure was explained by our Ramachandran in the name of triple helical structure. This much enough. After that, some more information you have to learn about Linus Pauling. Linus Pauling was the scientist who worked in Cambridge University. By inspiring the Linus Pauling work, our Ramachandran discovered triple helical structure of collagen. What he did? He explained secondary structure of proteins. Protein secondary structures. What are those protein secondary structures? This alpha helix and beta pleated sheets. Alpha helix and beta pleated sheets. These alpha helix and beta pleated sheets also secondary structure of protein. They were discovered by Linus Pauling. But the conformational biology that was applied by the Linus Pauling could not be explaining triple helical structure of collagen. The triple helical structure of collagen was explained by our G.N. Ramachandran by using a concept called Ramachandran plot. This much is enough for us. Associated with the scientist, remaining things, where he was born, what are the educational qualification, and where did he died, those contents they may not going to ask. Anyhow, the Ramachandran concept is enough. Then go to another thing, that is, why do you say cell is the basic structural and functional unit of life? We believe that cell is the structural functional unit of life. Why? What are the conformationals that you can able to provide that cell is the structural and functional unit of life? For that, some statement. Anything you take less than complete structure of a cell, complete structure of a cell, anything less than complete structure of a cell does not ensure independent living. What you are targeting on this statement? What we are trying to explain? Simple. Imagine if something you wanted to say as living. First and foremost thing, it must be having at least a single cell. If there is a living organism that does not possess single cell even, that thing should not be considered as perfect living organism. This statement they might be generated by observing viruses. You know viruses, they are cellular bodies. They are the connection between living and non-living. As long as the virus present outside the living organism, it exists as non-living matter. Once the virus has penetrated inside the living cell, it started to show living properties like growth, reproduction, and some amount of metabolism. That clarifies. You wanted to behave like a living thing. At least you must be having one complete cell. Anything less than a complete cell does not ensure independent living. Next one. Another definition to ensure cell is the basic structural and fundamental unit of life. A single cell can perform all the metabolic activities or all life processes, you can say all life processes, which means you consider reproduction, you consider cell division, you consider anything you can do that would be possible by the development of a single cell. That's why, since a single cell can perform all the life processes, and at the same time that anything less than a complete structure of a cell does not ensure independent living, so that we will say cell is the basic structural and functional unit of life. This is the most commonly asking assertion and reason type of question. Why do we say cell is the basic structural and fundamental unit of life? Reason: anything less than complete structure of a cell does not ensure independent living, or a single cell can able to perform all life processes. If you wanted to see a life developing, at least there must be a presence of single cell. Then here one thing, animated means living organisms are also called animated bodies. Animated means something that it can able to show movement by itself. Living organisms only can show move. That's why living organisms are called animated bodies. Next one, inanimated means non-living. Living and non-living, be careful, these definitions we will get continuously. This is an inanimated feature, this is an animated feature, like that they could guess some kind of informations there. Wherever you come across animated feature means living one, inanimated means non-living. But one thing, viruses, we will get about them later. Viruses, connecting link between animated and inanimated features, inanimated bodies, which means there are some properties of viruses we could see in inanimated bodies, that means non-living bodies, and there are some features of viruses could be noticed in animated. So inanimated bodies would be connected at one area, that name is called viruses. This about why do we call cell is the structural and basic unit of life. Next, go to cell theoretical concept or cell theory. Cell theory, you know this, the cell theory was proposed by two scientists together. One person is called Sleeden and another person is Schwann. In 1838, a German botanist named Theodor Sleeden, what he did? He is given one extraordinary statement like this: all plants are made up of cells. This statement he was given in the year 1838. Careful, this statement may not take much importance now, but on those days in 1800 beginning, this statement was very intellectual concept introduced in the cell biology, because people used to believe plants are dead. They do not have living characters. But this person said plants are having some sort of cells, because already the concept of cell was given by Robert Hooke and Antony van Leeuwenhoek in 1600s. Now in 1800, he said plants made up of cells. Remember one thing, I think you know one scientist named called Jagadish Chandra Bose. He was the person officially announced plants are living bodies. Before him, they used to believe mostly plants may be dead because they don't have all kind of living properties. But Jagadish Chandra Bose was announced officially plants also living. They do perform all living activities. They are a kind of living organisms. Official denotion. But on those days, at the time of Sleeden, he just mentioned like this: I have observed many plants and their organs. Then it says plants made up of cells. Okay. But in the next year, and at the same time, 1839, one more scientist came. He was also German, but he was zoologist. This one you should be very careful. In old NCERT book, it is written as British zoologist. About Schwann, it is written as British zoologist, but actually he was also German zoologist. He studied many things. Most of the contributions associated to cell theory was given by Schwann. What was those contribution? He given some statements like this: all animals are also made up of cells. Also made up of cells. All animals made up of cells. Same. Somebody given all plants made up of cells. Similar to that, I have observed this. In animal tissue also there are cells. So all animals also made up of cells. Concept given by Schwann. And he said one more: animal cells contains an outer boundary. Nowadays it is called plasma membrane. See the thing. First time Schwann said this. When I was observing some animal cells, I have noticed one thing. Animal cells are having some kind of outer boundary. This is the first time somebody said cells are covered by something. It's not about the significance of discovery of plasma membrane, but it says like this. Somebody right now is saying animal cells are having some kind of outer boundary. They are covered by some sort of boundary. Before him, there were no concept of boundaries present around the cells. Then he announced that there is a presence of an outer boundary around the animal cells. That outer boundary, right now we are calling it as plasma membrane, but he was not given the name called plasma membrane. Okay. He said finally, animals, or one more statement about him, presence of cell wall is the unique feature of plant cells. Presence of cell wall is the unique feature of plant cells. Why this statement come? Listen, if you search about cell walls right now, it's not that cell wall will be present only in around plant cells. Bacteria have cell wall, fungi will have cell wall, and some of the protista like dinoflagellate, diatoms will also having cell wall. But why did you say here presence of cell wall is the unique feature of plant cell? On those days in 1800s, this was the information available. They know only few cells. Out of them, animal and plant cells were very famous. Plant cell has cell wall, animal cell has no cell wall. Then he said, presence of cell wall is the unique feature of plant cell. Simply, he could see two cells. One is an animal, another one is a plant. Plant cell has cell wall, animal cell has no cell wall. So he said, presence of cell wall is the unique feature of plant cell. So with all these explanation, finally he gave one statement, that is, both plants and animals made up of cells. Both plants and animals, both plants as well as animals made up of cells. Who gave this statement? Same, Schwann. I have studied plants and animals, both are made up of cells. Very good. Then in one fine day, they have formulated cell theory. Both Sladen and Schwann together formulated the cell theory. What this cell theory exactly says? The cell theory that was given by Sladen and Schwann together. They say all living organisms made up of cells and products of cells. That's it, nothing else. All living organisms made up of either cell or something that it has comes out of cell, nothing else. Imagine, let us consider our body. In our body we will have, cells. In addition to that, bones. Do you believe we have got these bones from somewhere else? No. There are some special cells in our body called osteoblast. These are the cells, they have secreted some chemicals known as calcium phosphate, and that is the place where there is a formation of bones. Similarly, we have hair. Are there cells? No. But the keratin material secreted by some specialized cells. So, final conclusion, anything that you can see in a cell or anything that you can see in a body of a living organism, either it is made up of cells or some kind of material that would be comes from the cell. This is the concept which was there in first days of cell theory. But they could not explain something that it is, how did new cells arise? How did new cells are coming? Why the concept of how did new cells arise? Simple. Whenever we talk about multicellular organism, one cell is not present and at the same time all the cells are same type. No. More number of cells and variety of functions they are conducting. Okay. In this thing, carefully observe. Whenever you talk about a multicellular organism, its life is started with a single cell called zygote. So, how did all these cells came? Slidden and Schwann could not explain. That simplifies this. There is one scientist called Rudolf Virchow. Rudolf Virchow. What is this Rudolf Virchow? In 1855, he said one statement, that is, omnius cellula acellula. Omnius cellula acellula. In their language, this is the concept. It's actually Latin phrase. Omnius cellula acellula. It says like this: all the cells arises from pre-existing cells. Pre-existing cell. No de novo synthesis of cells. There is no what? De novo synthesis of cells. There is no concept of de novo synthesis at all. There is no cell can be synthesized in an activity of de novo synthesis. What do you mean by de novo synthesis? No cell can be constructed newly. If you want to get a cell, you can get a cell from already existing cell. During the course of evolution, there was a development of first cells. From there only remaining all cells arise. That signifies there is no new construction of a cell. You cannot construct a new cell. Since you already know plasma membrane, cytoplasm, nucleus, Golgi complex, variety of cellular components present. Collect all those components, you cannot construct a cell. You wanted to have a living cell that would come only from already existing cell. That's the reason why there was a development of a concept by Rudolf Virchow, that is omnius cellula acellula. Cells arises from pre-existing cell. No cell can be newly synthesized. With that, there is a final shape of cell theory. Final shape of cell theory. What is this final shape of cell theory? The final shape of cell theory, actually, final cell theory was given by Rudolf Virchow. He said, all living organisms made up of cells and their products. All living organisms made up of cells and their products. Next one. All the cells arises from pre-existing cell. Pre-existing cell. Which means no cell can be newly synthesized. Every cell can be arises from already existing cell. One moment. Okay. This is about final shape of cell theory. When it comes to final shape of cell theory, listen the statement. Whenever we talk about living organisms, all the living organisms arises from pre-existing one. This applicable to both cells as well as living organism. No living organism just now newly constructed. Okay. Now, whenever we talk about this cell theory, the final shape goes like this. But one thing we need to be very careful about it. That is, there is some chemical in our science recently developed called viruses. Viruses. Cell theory cannot apply to viruses. You cannot apply cell theory to viruses. What is the meaning of that? Observe the word. Cells will have some fundamental features. Those features we cannot see in viruses. That's why viruses, since do not have any kind of cellular construction, majorly they are acellular in nature, acellular bodies. That's why cell theory you cannot apply to viruses. Apart from viruses, every living organism can show cell theory. Got it? This about cell theory and its details. Very important. If they wanted to get any one particular variety of question instantaneously in the beginning of cell and the unit of life, they will search in cell theory. Okay? Then after that, overview of cell. You know, there are some cells which are in the name of eukaryotic cells, which are having well-defined nucleus, and there are some cells are called prokaryotic cell with defined nucleus. Nucleus is properly seen in these cells. They are called eukaryotic cell. Now I am talking about details of eukaryotic cell first. There is a presence of eukaryotic cell. If you see, they have cell wall, and in that protoplast. Carefully follow how I am dividing. Then this contents later you will have clarity while we are studying. Different places you will see there is a cytoplasm, there is a cytosol, there is a protoplasm, there is a protoplast. Like that words you will come across. So this is the time to learn the fundamental differences between cytosol and cytoplasm, protoplasm and cytoplasm, etc. Okay? Then I am talking about a eukaryotic cell. I am saying eukaryotic cells will have cell wall. Do all eukaryotic cells will have cell wall? Of course not. But some eukaryotic cells we will see cell walls like plant, fungi. Next one, there is a presence of some cells like dinoflagellate, diatoms, chrysophytes. All of them will have cell wall. But animal cells do not have cell wall. That concept we know. Then come back to what is protoplast. When you talk about protoplast will have two things. Or else in a cell, I will remove cell wall. The remaining area is known as protoplast. The protoplast will possess plasma membrane. We will talk about it. Sometimes it's also called cell membrane. Next one, protoplasm. Plasm means fluid or some kind of soup-like material. Protoplast consists of plasma membrane, and the next one is called protoplasm. Observe, the protoplasm will be having again cytosol or cytoplasm, in the protoplasm. And one thing, in protoplasm, this side I will write the less number. It consists of nucleus or nucleoplasm. Next one, cytoplasm. Plasm means fluid or a kind of soup-like matter. Nucleus. Observe. Nucleus contain nucleoplasm, and also inside the nucleoplasm, plasm means fluid. Inside the nucleoplasm, there is a presence of chromosomes. The chromosomes contain DNA. Of course, the DNA is the genetic material. Here I wanted you to remember one thing. Wherever hereafter you come across a word DNA, apply DNA is nothing but chromosome, vice versa. Chromosome, if you have seen, this is talking about DNA. In future I will differentiate what is this DNA and what is the chromosome relationship, but right now chromosome is nothing but DNA. Of course there is a variation, but timingly here remember chromosome means DNA. Now in the nucleus there is a fluid portion called nucleoplasm and the chromosome which are carrying genetic material. That much simple. Next one, cytoplasm. When you talk about cytoplasm, you will see cytosol. Cytosol. This is just a fluid. Cytosol. The next one, cell organelles. Cell organelles. Listen, what is this? I have taken cytoplasm, which is a soup-like matter, and I have filtered. Some liquid will come out, some solid substances will remain. What are the solid substances will remain? They are called cell organelles. What is the fluid that can pass through filter? That fluid is known as cytosol. Cytosol is a fluid matter. Okay? Now go to cell organelles. When we talk about cell organelles, you can take the cell organelles into two categories. One is non-membranous. Non-membranous. Could you tell me what are non-membranous cell organelles that you have faced? Majorly ribosomes you have seen. Ribosomes, which are universal. Wherever there is a cell, you will see ribosomes. And again centrosomes. Centrosomes are particular. Centrosomes could be seen only in case of animal cells. Anyhow, the centrosome and the ribosomes do not have membrane. Okay, they are non-membranous. Then what about other category? Membranous bodies. These are membranous. The membranous ones will be having again single membranous. Next one, double membranous. Double membranous. Could you tell me double membranous which is easy for us? Mitochondria and chloroplast. Especially plastids you can come, but here I am writing chloroplast. Next one, single membranous. We do have among the single membranous endomembrane, non-endomembrane, etc. That distinction I will give you later, but right now single membranous you can count many. Endoplasmic reticulum, Golgi complex. Okay, Golgi complex. Next one, lysosomes, vacuoles, peroxisomes, etc. Many are there. Some I have mentioned here as an example. Like this, many things will be present inside a eukaryotic cell. You see this chart development, how I have taken. Eukaryotic cell, some cells may have cell wall. Remaining part is known as protoplast. Protoplast will have plasma membrane and protoplasm. Within the protoplasm you have nucleus and cytoplasm. Within the cytoplasm you will have cytosol and organelles. Among the organelles you will have membranous and non-membranous. Non-membranous somewhat rare, membranous very common. Among the membranous you may have again single membranous and double membranous. Whatever you take, you make like this a particular chart. Among the chart you just notice there are some exceptions, etc., etc. In future these kind of charts are very useful for making the things to easily recall. Okay, this about eukaryotic cell one. Now I wanted you to think examples of eukaryotic cell, what you can give. They are asking, give some examples of eukaryotic cell. By chance, if they say, what are the examples you can give to eukaryotic cell? Simple. What are they? Imagine. Over there, you can say protista members. Next one, fungi and plants and animalia. In that, I have seen many students are getting confused. Protista. In the protista, we have chrysophytes, dinoflagellate, slime molds. Next one, euglenoids and also protozoan protist. Among the protozoan protist, you will see our wonderful unicellular eukaryote that's called amoeba. Keep it in mind, yes. I have observed many students getting confused about amoeba and E. coli. E. coli is a prokaryote, which is a bacteria. Amoeba is a eukaryote, but even it is eukaryote, it's a unicellular eukaryote. That distinction we will talk about later. Then go to prokaryotic cell. What is the overview of prokaryotic cell? One thing, wherever you come across a word prokaryotic cell, that signifies you are talking about bacteria. What is bacteria? Imagine, when we were talking about a kingdom Monera, there you said bacteria are the sole members of the kingdom Monera. What do you mean by the soul members? That's not that soul, S-O-U-L, Atma, Paramatma, no. That soul is S-O-L-E, soul. Soul means single, alone. In the kingdom Monera, only one type of organisms present. They are called bacteria. Bacteria, the sole members of the kingdom Monera, which means within the kingdom Monera, only one type of organisms are present, which are called as bacteria. Observe, in that case, bacteria, varieties are there, eubacteria, archaebacteria, some names I will denote. First one, cyanobacteria. Cyanobacteria. One more is there, mycoplasma. I think you know, mycoplasma is the smallest living cell in the entire living world. So far, among the cells which are noticed, the smallest living cell is nothing but our mycoplasma, which is around 0.3 micrometers. Next one, it does not have cell wall anyhow. Next, go to rickettsia, chlamydia, spirocheta, etc. Many will be there. Many types of. One thing you remember, all can be called as bacteria. If anybody says mycoplasma is a bacteria, right or wrong? Of course, correct. Mycoplasma is a bacteria because it is present in the kingdom Monera. All prokaryotes you can call default state as bacteria. Okay? Next one. One more category I have forgotten to write, that is archaebacteria. Now, take a prokaryotic cell and its detail. Overview of prokaryotic cell structure. Now I want to define what is the overall construction of prokaryotic cell. First one, we need to know, if I want to call something as a cell, what are the components that cell must possess? Observe, you said cell is a compartment. Good. In that compartment, first of all, there should be a presence of an outer boundary, isn't it? That outer boundary, what do you think? A cell must contain an outer boundary. What do you think the outer boundary of a cell? Of course, it should be plasma membrane. Plasma membrane, never be cell wall. Why never be cell wall? Cell wall is not universal. Some cells may have cell wall, some cells may not have cell wall. Do you think that the cells which are not having cell wall do not have boundary? Of course they have boundary. That boundary is nothing but our plasma membrane. Plasma membrane also called as cell membrane. Exactly to say, cell membrane is known as plasma membrane. What is the difference? Around the cell there is a membrane, you can call it as cell membrane. But that cell membrane has a name called semi-fluid in nature or quasi-fluid membrane, etc., etc. Because of their quasi-fluid or semi-fluid nature, this membrane is known as plasma membrane. Plasma, a status of a physics which is existing between the solid and the liquid. Solid, liquid, gas, okay, we have seen these are the physical forms of the matter. But some of the physical forms of the matter existing between solid and liquid. Solid and liquid between the matter is known as plasma. So no need to confuse. You can take, there is a, look at the statement. There is a semi-fluid matrix or there is a plasma, quasi-fluid membrane will be present. That quasi-fluid membrane is known as cell membrane or plasma membrane. Clean the board. Next, go inside. This is next main components for a cell. This area is known as cytoplasm. Cytoplasm. What is cytoplasm? Cytoplasm is the main arena. Arena means area, ground. Main arena of cellular activities. Cellular activities. What do you mean by this? Whenever we see a cell, keep it in mind, that cell must contain two things. Remaining things present or not doesn't matter. That cell must contain two things. What are they? One is called plasma membrane, another one is called cytosol. Remaining all are extra decorations. Sometimes a cell may develop cell wall, sometimes a cell may develop flagella, sometimes a cell may develop cilia, some cells will have nucleus, some cells will have cell organelles, some cells do not have them. Okay, but finally one thing, you wanted to call something as a cell, two things should be there. One is plasma membrane, outer boundary. Inner area is known as cytoplasm. The cytoplasm is the semi-fluid matrix, semi-fluid area matrix, that is called cytoplasm, which is also called the main arena of all cellular activities. I wanted to kill a cell. Cell is living, be careful. Cell is living. I wanted to kill a cell. What I have to do? Break its plasma membrane. Cell will die due to leakage of cytoplasm. Like that, all cells will have two things in common. One is plasma membrane, another one is cytoplasm. Remaining all are developed depending upon the requirement of a cell while it is modifying into different forms. Since we are talking about the word called modifications of the cell, observe, different cells will have different structure, different shapes. Simply, structure and shape of the cell depending on the function that it performs. One more time. Cells will have different shapes, different sizes, different structures. All these modifications are developed depending upon the function the cells perform. For example, RBC possess biconcave disc-like structure. That is purpose, not decoration purpose. Just purposefully the RBC have become biconcave disc-like shape because that shape will make them to absorb maximum amount of oxygen during at the time of transportation of it. Okay, nerve cell, which is called a longest cell. While nerve cell is longest because it has to connect from one place to another place in order to send nerve impulse. Next, like that, different cells will have different structures. Some cells are columnar epithelial cells, some cells are cuboidal epithelial cells, some cells are long tube-like cells. Okay, cell structure depending upon the function that it performs. One thing they have given, the longest cell in the body or longest cell in the nature. nerve cell. The largest cell is isolated ostrich egg. What do you mean by isolated ostrich egg? It's not that the egg that was laid by ostrich, which is also large size, that's not they are talking about. Go to the ovaries of ostrich and there you will find some kind of eggs. Egg is a cell, isolate it, then you will get the largest cell. That's not the omelette that is given by the ostrich egg. That is, for example, if ostrich has laid a shell, calcium carbonated shell coated egg, that egg is actually embryo containing body. Of course we call in regular terms that is an egg, but actually it's not egg, it carries an embryo inside. Embryo is not a single cell, but if you wanted to see a single largest cell, go to the ovaries of an ostrich, it can produce a largest cell in the living world. Then go to overview of a prokaryotic cell. If you take a prokaryotic cell, first of all, it has cell membrane. It must be, as I told you, without cell membrane, there is no something called a cell. Around the cell membrane, there would be a presence of some material that's called cell wall. Do all prokaryotic cells will have cell wall? No, there is an exception in the name of mycoplasma. Cell wall. Which prokaryotic cell without cell wall? Cell wall. That is called mycoplasma. Okay. Go inside. This is plasma membrane. Inside, cytoplasm. Inside the cytoplasm, prokaryotic cells generally possess single circular chromosome. This single circular chromosome means DNA. The DNA always double-stranded DNA. One more concept, fundamental thing, you have to learn this. Wherever I see DNA, I will always talk about double-stranded DNA. Default, if I say there is a DNA, you should believe this DNA is double-stranded. If I talk about single-stranded DNA, I will notice it specially. There is a special kind of viruses which are having single-stranded DNA. Unless we notice there is a speciality, DNA would default indicate this is double-stranded one. Don't confuse. Single-stranded DNA is never present in cells. You take a prokaryotic cell, you take a eukaryotic cell, animal cell, plant cell, fungal cell, okay. But there is no single-stranded DNA in them. All of them possess double-stranded DNA. Next one. When you talk about viruses, some viruses here and there who are having single-stranded DNA. Unless that say, wherever you come across, there is a DNA. Default state, you believe this is double-stranded. If they say only, you believe it is single-strand. Otherwise, it is double-strand. So, I am saying it is single-stranded circular chromosome. The single-stranded circular chromosome is called naked DNA. Naked DNA. Why did you say it is naked DNA? Because this DNA has not covered by any sort of nuclear membrane. That's the reason why we call it as naked DNA. The naked DNA has another name called nucleoid. Nucleoid or xenophore. Their names and their data, later we have elaborated discussion. Just I am saying there is a DNA in the center. That place is called nucleoid, which is naked because it does not covered by nuclear membrane. Next one. Some bacteria possess extra chromosomal circular DNA. Circular DNA. One more word I want you to be remember perfectly. Perfectly. Okay. Wherever you see prokaryotic DNA, in default state prokaryotic DNA is circular. Eukaryotic DNA is linear. I'm not saying all prokaryotes will have circular DNA. That's a difficult statement to your level to understand. But whenever you come across prokaryotic DNA, whether I say it or not, you should believe it is circular. If it is linear, I will inform to you. But default state it is circular. Now, main chromosome is this. This is extra chromosome. Small, minute, cute DNA will be present in some of the cells. That DNA is known as plasmid. Plasmid not for all. Okay, plasmid only for few. In that case, take a plasmid which is so-called extra chromosomal circular double-stranded DNA. All bacteria it is not belong. So some bacteria wherever it present, it gives unique phenotypes. Some sort of unique phenotypes. What do you mean by unique? I have a bacteria without plasmid. I said all bacteria do not have plasmid. I have a bacteria which has plasmid and I have a bacteria without plasmid. I can easily distinguish by the phenotype that bacteria shows. What are the phenotypes I will catch? I will catch some phenotypes like antibiotic resistancy. Antibiotic resistancy. Now, what is antibiotic? You know, some antibiotics kill bacteria, or else bacteria killing substances are known as antibiotic. See this one. We have seen that word ampicillin, tetracycline, kanamycin, erythromycin. So many antibiotics are there. Antibiotic, take few drops, keep it on a bacteria. That bacteria dies. That bacteria does not have plasmid. Next one. On another bacterium, you have kept few more drops of same antibiotic. That bacteria survived. This bacteria has plasmid because plasmid is something that could be providing unique properties. Among the unique properties, one unique property is called antibiotic resistancy. Antibiotic resistancy a bacteria will get only with the help of plasmids. I am not taking all plasmids will provide antibiotic resistancy, but whatever the bacteria has got antibiotic resistancy, that antibiotic resistance is due to plasmid. Keep it in mind. And also, these plasmids are responsible for transformation. Responsible for transformation. I think when you were studying about second year, you have come across one person known as Griffith, and he had identified Streptococcus pneumoniae in the name of transformation. What is transformation? Sending of the genetic information from one bacterium to another. That information sending is possible only with the help of plasmid. That means one bacteria gives information to another bacteria. Getting the idea? One bacteria giving information to another. How do they share the genetic information? By through mobile phones? No. There is a mechanism existing there that is called transformation. That transformation is also happen only with the help of plasmids, nothing else. Okay? Is it okay? Next, like that many properties. One more thing. Some bacteria possess mesosomes. Who are mesosomes? What is their story? We will have the big story. And some bacteria possess chromatophores, etc., etc. Chromatophores. And some bacteria possess some kind of unique component called inclusion bodies. They are storage granules. Okay, this is the overview of a bacteria or a prokaryotic cell. Do you have any queries, objections? You can say there, I can explain. That's about today. Up to this, you will have exam on Sunday. Come on. Anything else you need, I can help you. Don't study cell and its envelopes, prokaryotic cell envelope. Is my words are clear to you? Are you able to follow me? Anything required? I can project in front of you. Am I clear? Come on, I'm still with you. Yes, this chapter is also there for exam. Come on. Anything else you like? Okay. We'll meet in the next class. Bye for you now. Okay?